We have an important component, which is food, right? Curing through medicine, food. Oh, we're already over. Good morning, everybody. Welcome to the Spring 2023 Land Grant Seminar Series. I think we're good to go, right? I just want to take this opportunity to make a few announcements. The first one is about our graduate degree program in natural resource and environmental sciences. We have the master's degree program. I have a couple of flyers there. One of them is about our MS degree program. Please pick one of these two. I have another one, but pick one and read through it, and most importantly, share it with your colleagues, and most importantly, share it with potential graduate students who we would like to recruit to be part of this program. I'm going to give you a quick overview of our graduate program. It's to promote and protect our natural resources, advance your professional and fellowship and follow your passion. So the professional program provides a pathway for manifesting careers in academia, teaching, and research, and also applied research and extension in education and governmental institutions at the national, international level, and also policies, and if you are interested also in policy. The curriculum provides a thorough background in research opportunities through very timely topics like climate change, water, energy, food nexus, soil health, watershed hydrology, geospatial tools like GIS, remote sensing, and natural resources economics, to cite a few. Admission, we are very, our admission is not stringent. We just need to have a GPA higher than 2.75, TOEFL of, if you are an international person who English is not the mother tongue, you need to have 550 in paper test or 79 internet-based scores. Selected ongoing projects, we have very few projects, to cite few, and these are from the core faculty like Dr. Rupendra Awal, who is here, Dr. Ramray, Dr. Peter Ampim, myself, and Dr. Richard Griffin, and others. So we do have few projects that are really related to that area. Another announcement that I would like to make is we are having a conference on water, climate, and food security for students and early career scientists. It's going to be March 6th through 10th. The first 3 days, 6th through 8th, will be for a training in 3 softwares, IPEX, FarmSim, and SWAT. So we are accepting applications to attend this. More importantly, we would like our students to participate in this conference. So Dr. Carson, I'm sure reached you, but I also would like to remind you to consider supporting your students to present in this conference, and also for our early career research scientists also to present. And this is a great opportunity for us to showcase of Prairie View A&M University, because we're going to be having students and junior faculty, as I mentioned, from the 1890 institution. Imagine it's a mini ARD symposium. So it's like, I'm sure all of you have been at, most of you have been an ARD symposium. You can see this one as a mini conference like that. So we're going to have it here, and your support is highly needed, especially encouraging our students to participate. And you see the topics there, or the themes. The conference is 5 themes. We'll send an electronic copy of this announcements. So go back to the spring semester outline of the presentations for the seminar. The slots are filling quickly. We only have a few left, so I encourage you, if you are interested in having someone talk on your behalf, or you would like to talk, we encourage you to reach out to Dr. Awal and tell him about your interest. I think before I go there, before introducing our speaker for today, which is should be page up. Okay. I had a flyer here about our REEU, Research Extension Experience for our undergraduate students. Please encourage your students to apply. We're still, Prairie View A&M University, we have up to 5 or 6 students. We didn't get enough applicants. So please spread the word around, take a copy of the flyer, encourage one of the students to apply. It's a great experience, 5 weeks, they get 2,500 stipend, free on-campus housing in College Station. They get $1,000 as meal allowance, in addition to the stipend. And also the travel allowance for a field trip when they go. And this year, we're going to take them also to spend a few days in Galveston too. In addition to being in College Station, they stop here for 1 day, and they also go to Galveston for a few days to test the water quality there. So encourage your student to participate there. Okay, now we come to, yes, Dr. Memley. Thank you, Dr. Erdogan. That's Dr. Awal shared that with Dr. Sanders, who's dealing with the clubs, but we will make sure that they receive that. We also shared it with our faculty, but it takes some effort from each individual to that this is something that we need to see our student take advantage of. And don't be, I mean, I think it's although the theme is about wastewater, but the techniques that they learn about microbiology, about chemistry, about all of that are relevant to any of the fields, even to pre-vet, because all those techniques, they would need them in their training. So today's speaker is Dr. Samah Ahmed. He is an assistant professor of chemistry in the College of Arts and Sciences across the street from here. If he opens the window, he can see the building. He, prior to Prairie A&M University, he was a postdoctoral research fellow at Cornell University, Ithaca. Dr. Memley, there is a link here, and the Department of Chemistry at Texas A&M College Station. Dr. Memley was also a postdoc in Cornell, or a visiting scientist to be correct. Instructor part-time at Blinn College here in Texas. His education, he got his PhD in organic chemistry from Marquette University, MS in medical chemistry from Cairo University in Egypt, and BS on Cairo University in Egypt as well. His field of interest is organic synthesis that impacts areas such as medicinal chemistry, materials science, and reaction mechanisms. He has several current projects. To cite a few, one is design and synthesis of novel electroactive molecules, nanotechnology, and biomaterial applications, and design and synthesis of bioactive organic compounds and applications of different chromatographic techniques for purification of products, structural assignment of the products, and evaluation of their biological activity. He has several accomplishments. To cite a few, he is well published with more than 30 peer-reviewed publications. With that, I would like to welcome our speaker for today, Dr. Samah Abdulwahid, to talk to us about design, synthesis, anti-proliferative, and computational studies of new coaxiline-based derivatives as PARP1 inhibitors. Dr.
Thank you very much, Dr. Faris. Thank you very much, Dr. Faris, for giving me this opportunity to talk to the agriculture department. So today I'm going to talk about medicinal chemistry. As Dr. Faris mentioned, I have some kind of different activity in chemistry. So basically, I'm making compounds. This compound could be biological active, could be using as semiconductors or any field of material science. But today I'm going to talk about medicinal science. So basically, as you can see here on the title over there, so there is something called quinoxalines. So it is a chemical compounds. And here in BriarView, I was able to design and synthesize this compounds, and with collaborative with with other faculties over Egypt, we did the biological activity, and with another guy in University of Philadelphia, we did the computational study. So let me, because I might know you guys don't have maybe good background about the chemistry. So I will try to make it as simple as I can. And perfect. Okay, great. Which is going to actually lead me to ask you if I'm open for any collaboration. So if you need any help in kind of what you are going to hear right now, so I will be really happy to have any kind of collaboration in future. So let me just give some outlines about what I'm going to talk about. I'm going to talk about cancer, okay? So I'm going to talk about cancer disease and what is the treatment. I'm going to talk about some facts about the cancer as disease. Also, I'm going to talk about something called BARB enzyme. So BARB enzyme is one target, which is the research focus on inhibiting this enzyme. And I'm going to talk shortly why we need to inhibit this kind of enzyme. I'm going to propose some compounds which actually help us to inhibit this enzyme. And I'm going to talk about characterization. I'm going to end with conclusion and acknowledgements. So, what is the cancer disease? So basically, why cancer happens? So cancer happens when there is some kind of, so the normal cell has a balance of two things, of the growth of the cells and the apoptosis, which is the death of the cells. So there is a balance between them. And if the balance change, so you can expect something happen, which is cancer. And as you can see, the gene which is controls the growth is called oncogenes, and the one which is responsible for the death of the cell, we call it tumor suppressor genes. So we are normal, hopefully. So you have a balance in the normal cell. If something happens in the genes, you can expect something wrong going on. So let's see why research is working with cancers. So there is some facts about cancer. The first one is, well, it is the second leading death in the world. And 10 millions die every year over the worldwide. And half, the first, well, good question. Maybe accidents, I don't know. Yeah, or maybe combination of other is heart. Maybe heart attack, yeah. Yeah, good question. So yeah, here in US we have about 500,000 every year die from cancer. Well, the good news is you have 40% of the people expecting to get cancer to avoid this. And avoiding means you are going to go like don't smoke, don't take alcohols, good food, and so on. So this is, you can just avoid it a little bit, but maybe because you might have kind of gene, you can't do anything about it, basically. Also, the other thing is 1/3, you can prevent also this by doing kind of screening, like you go to the doctor, making kind of gene check, all stuff. So you can try to avoid it before it happens. Well, the bad news is there is no cure for cancer. But cancer can treat you and can cure you. But for 100% cure, I think you need to take the drugs every time or cut the part which has the disease. So, so basically the treatment is surgery, chemotherapy, or targeting medication. Today, I'm going to talk about targeting medication. And basically, as you can see here, if there is a cancer cell over here, there is many ways over here that you can enter and interfere the cancer. Today, I'm going to focus about, as I said, it is PARP inhibitors. So it is, so somebody will ask me, what is PARP? So basically, PARP is enzyme. And this enzyme is polyadenosine diphosphate ribose polymerase. So what this enzyme doing is, this enzyme is going to search for any kind of break in the DNA and is going to heal this. So basically, this is the PARP enzyme is going to detect any kind of any single or double strain break in the DNA and is going to just put them together back. Okay, so basically what it's doing is going to repair the DNA. So why, so since this enzyme is important, so why people want to inhibit this enzyme? It doesn't make sense, right? So, but if you look at the diagram, which I have it over here, so why people want to make a medicine, make a compound which is going to inhibit the PARP enzyme, since it is important actually to cure the DNA. So let's look at the normal cell versus the cancer cell. So if you look at the normal cell and some break happen here in the DNA, there is two mechanisms to repair this. You can use the PARP enzyme to heal it down, like here, or to use the PARK. So PARK is gene, and this gene actually has responsible for also repairing the DNA enzyme, repairing the DNA. So in normal cell, you have these two mechanisms to repair DNA. And here is the idea. The idea in the, if you have any kind of gene mutation of this PARCA, and by the way, PARCA is, as I can see, BRCA, so it is coming from breast cancer. So this gene actually responsible for the breast cancer, responsible for ovarian cancer. In men, it is responsible for prostate cancer and so on. So in this cancer cell, well, this gene has kind of mutation, and this mutation will not make it available to make this kind of work, which is DNA repair. So what does it mean? It means like if we can just target the PARP inhibitor, as you can see here, well, since the human has this gene with no mutation, well, it is a good gene, so you can predict it's going to cure it. But for the cancer cell, if you inhibit the PARP enzyme, so it means there is no way to repair the DNA. So when you don't repair the DNA, so you are going to kill the cell. So this is kind of indirect way to kill the cancer cell. Okay, and this is why also many of the companies, they target the PARP. Okay, again, as I mentioned, there is many ways to kill the target cancer. This is the way which we are going to focus today. Okay, so as I said, there are many drugs available for inhibiting the PARP. So one of them, actually, which I'm going to focus on, so it is the commercial name is Lynparza. So Lin-Barza is a commercial name for this kind of structure over here. As you can see, sometimes it is kind of complicated structure. Maybe it will take many steps to make this compounds. So actually, we took this as our model to design our compounds. We took this our model to design our compounds. So of course, when you are going to design a molecule, you need to see the 3-dimension inside the active site. So any enzyme, as you guys know, has what's called active site. And this in the active site, all the chemical reaction happen. The DNA repair is going to happen. They design this compound, for example, to come in the active site of the enzyme and is going to inhibit. Is it going to just kill the enzyme? Like it's going to, this drug is going to come inside the enzyme and it's going to just block the active site, so it will not be able to do its function. As simple as this. So basically, there are many drugs approved with FDA. So I'm going to focus, as I said, on our study about this compound over here. There is some facts also to know about this compound. It is not chemotherapy or medication. It has very good actually effect, like about 67% of the patients. So is taking this medicine, they can be. Again, it is depend on the stage of the cancer. If it is in a very early stage, yes. But if it is in 3rd or 4th stage, it's hard to deal with it. The bad thing about this, it is really very expensive. You can, so for 60 tablets, you might pay this amount of money, 8,000. So actually, this is actually encourage us to do our research. So our research is to make another compound, okay, with low cost. And same time, we do it in kind of some kind of chemical steps, which we call it green chemistry. So this is what we focus in this one. Actually, I have published this work last year, end of the last year. So let's see how can we design the molecules, which really looks like the commercial available drugs. And it might, so let's see, it might have better activity. So again, when you look at the enzyme and you want to design any molecules, you have to look at the active site, the 3-dimension of the active site, and try to make a compound which is going to inhibit this enzyme. Okay, so the compound is going to come in the active site. It will make some kind of bonding. Okay, mostly it is hydrogen bonding in the active site, and it will inhibit the enzyme for its work. So the molecule which the enzyme used to repair the DNA is this molecule over here. Okay, so it's part of it is nucleotides, nicotinamide part over here. This is phosphate part, and this is the adenosine site. So in the active site over here, you can find this molecule here. So let's look at the active site, and let's look at the active site also of the well-known crystal structure of the commercial available drug. So this is the commercial available drug, as I said, which is going to be our model. It's going to be our model. It means we are going to design a molecule which looks like this one, okay, with different 3-dimension. Hopefully it's going to be more active, and hopefully it's going to chemically we can do it with much easy, low cost money. So again, for the 2 dimensions, so here is this molecule here in the BARB enzyme. This is the 2-dimension. We looked at all the amino acid which is interact here with the compound. This is the 3-dimension. So basically, we looked at what's called computational study for this molecule, and based on this, we start to think, well, can we do something which is going to be compete with this one over here? And we came again with using computational study. We came to have this kind of molecules over here. Okay, and also as you guys know, the structure relationship, activity relationship study for the enzyme, we got to even more very accurate 3-dimension molecules. And in this slide, actually, here, I'm telling you the end of the story or the results. I just prefer to do this before I talk about chemistry, which is the part which I actually did to make this compound over here. So what you have to know is IC50 of this compounds is 4.4 nanomolar. Among about 30, IC50, it is yeah, it is half concentration, which is going to kill the cancer cell. So you need this amount of concentration to kill the cancer cell.
And R is radical? Radical?
The I?
R.
The R, sorry, R is it could be a hydroxy group, it could be methyl group. Yeah. So basically what you see over here, so this is R group here as derivatives. So for example, this one R, this is OH over here, and here's the methyl group over here. So this compound has this kind of activity over here, and for this one R is equal R dash means you have here methyl group and here methyl group. In this one you have R, which is well, it's going to be oxygen over here, OH, and here OH. Yeah, it's function group. It is a function group, yes. Yeah. So among these compounds, actually, and compare to the activity of the compounds, you can see here some of the compounds actually show a very good activity, like for example, this one. So you need this kind of very little concentration compared to the commercial available one to kill the cancer cell, which is really a good result over here. We have another compound, yeah, this one, when the R group is methyl group and the other one is methyl group here, it is 3.05 nanomolar. So this is, again, a very good activity. And the rest of the compound, actually, it doesn't like, looks very active like this one, but it is competing with this one. So by this way, and as I said, this can summarize all our work, which I'm going to talk about. How can we make this compound, which is right now showing like, yes, we can design and make a compounds which compete with the commercial available expensive compounds. And of course, we looked at the interaction of this active compound in the active site of the barb. Here is the 3-dimension. And again, all this work we have published last year. So maybe this is the area of what I'm doing here in Bright View. Since sometimes the facility here is limited, so this computational work, as I said, I have collaboration with someone in Philadelphia, and all the biological study, it was done in Egypt. So, but this is done here in the department. So basically, this is making this kind of molecules. As you can guys, if you can remember, this is one of the most active one. Okay. So let's see, for example, how can we make this compound? So you start with very simple compounds. So this is oxalic acid with orthophenylene diamine. You put them in acidic medium and you get this nucleus. And this actually what we call it quinoxaline. So this is quinoxaline, and why it is 2,3 because you have dione here and dione here. So we call it quinoxaline 2,3 dione. Okay, and this is a derivative of this one. So I are going to build some kind of function over here. Again, why we build this? To do two things. First, to make sure it's going to fit in the active site, and it's going to make kind of hydrogen bonds in the active site, so it will inhibit the enzyme for its activity. Well, second step is to put chlorosulfonic acid to put this group over here, then hydrazine hydrate to put this group over here. And from here and here, we make different kind of reactions. For example, here we treat this with acid anhydride. For example, if you treat this with succinic anhydride, you are going to close this ring. So you can AR here is this group. Okay. If you reacted with maleic anhydride, you are going to make this compound. If you reacted with phthalic anhydride, you are going to make this compound. Again, we have kind of library of these kind of compounds, and maybe more of this also I didn't show. And we screen all of this stuff and compare it and take the control of the well-known commercial drug, and we compare our activity to their activity. Here, you are going to react this with ethyl acetoacetate. Okay, to give this one, you react this with dimethyl malonate to get this one. You react this with acetyl acetone to get this one. Again, this is one of the most good active molecules, which we get very good activity. Moreover, we make a bunch of the compounds also. We'll start with this one, which I just talked about it before here. We make another bunches of compounds. So basically, we react the hydrazone part here with isothioacetate, different isothioacetates. So one of them is para-methoxyphenol group over here. Here is going to be with benzophenone, okay? And we can make kind of closure. It is either you can close the ring over here, or you can close the ring over here. So you need some kind of chemicals to make this cycle closure. For example, if you use malonic acid, you are going to close over here. If you use chloroacetone, you are going to close from here, and so on. You have here 3-chloroacetylacetone close over here, and you have the ethyl bromoacetate to close also over here. So again, we have a bunch of molecules based on our vision of how the active site C dimension looks like and what kind of bonding with amino acid we can create. And this is going to determine how good inhibition this compound is going to do. So as I said, we were able to make this kind of compounds, which has a very good activity actually compared to the commercial available drug, with like few steps and with, I will call it like cheap compounds. Yeah, so this is my talk for today. So I really thank the Office of Research for like they actually started with Dr. Faris in my first year when I came here. So actually I was lucky to get a good grant to establish my lab. Otherwise it will be very hard to initiate my work. I thank also Dr. Manal from Egypt They did the biological part and all this kind of uh, IC values and all stuff. Dr. Khalid, he is from Temple University in Philadelphia. He did the computational study and he figured out what good molecules that we can make. And as I said, here in BriarView, we make this compounds available. Okay, thank you very much. Thank you very much. Thank you, everybody.
Thank you, Dr. Abdulwahid. So the floor is open for any questions that you might have for our speaker of the
Sure.
day. We have a question that is related to me.
Yes. So, and I will ask the biologic side of things, how do you have the different derivatives, right?
Right.
How do you assess their competitive binding with other molecules and cross-reactivity and their effect on other biologic functions beside the function of inhibition?
Yeah.
That point, how do you do that? I mean, I understand your, I don't understand, let me rephrase that, but I gather with your modeling, it makes sense and everything. So how do you take that then to the biologic activity? Because, you recommended these two are the most promising of those. Have you looked at, and is that what the people at other institutions are doing?
Yeah, they looked at, actually they did it in vitro and in vivo as well. So they did study, to be honest, I don't know exactly what they did. The other part from your question, if this compound is going to affect on other thing.
Biologic systems.
Yeah, so the answer also, I don't have the answer because, when you make any drugs, it's going to go with first phase, second phase.
Right,
third
no.
phase.
And I do know that a lot of cancer researchers are moving
Right.
into modeling.
Right,
And that's why I'm asking if you can take your computational modeling here and move it into biologic modeling that is available today to see if there are predictive values of affecting other biologic systems.
sure, it will be a good idea to go further with these compounds. I'm
Okay.
sure.
No,
yeah.
no, that's okay.
Yeah.
I have another question here. Dr. Carson.
So, thank you. Thank you for your talk. I appreciate it. A couple questions. On your modeling, what program did you use to model?
In what?
In the modeling of your compounds and looking at the interactions,
Right.
right? What program did you use to develop your models?
Well, it is again good question. I don't know exactly what kind of so they did the computational with the software.
Right.
I might like I have it in the paper for sure
Okay.
because
okay.
we published this thing.
Because
But
I know.
what is the name of the software? I don't know exactly, but there are many kind of software available like Schrodinger, and this is the one which came to my mind.
Okay. Okay.
But of course there are many several. Actually, one.
But I know
one-
that there's some that's free. You can just download a whole series of them. and they're free.
The free software actually, because I think I did this one time like 3 years ago.
Okay.
It is very slow.
Yeah.
And at the same time, it is not going to give you an accurate results.
Right. Okay.
Because the one which they have, actually the university has this kind of facility and he runs this for overnight, something like this. And they even don't have the window. They have what's called Annex. So another computer system to run the
Yeah.
Yeah.
They have that database, I believe, at UT Austin, right? Do you go through
Well,
that
in UT
database?
Austin and Texas A&M, they have for sure, yeah.
Oh, okay.
But this is
okay.
the guy who I know, so
Yeah.
I told him to help me with this. Yeah.
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 So okay. I noticed that in your reactions that your yield is continuously decreasing, right?
So it depends on the like you do the reaction, right?
Right,
And you see how much you have.
right,
Is there any reason that you have low yield or high yields? Maybe, but our target actually is not to run to improve the yield more than to make the compound.
right. Okay, okay.
So yes, some yields is high, some yields is not. But I think all of them in a good range, I will say, like 60,
70 or so.
70.
That's pretty good yield.
Yeah,
actually.
it is still good yield, yes.
Yeah. Okay. So I guess my other question is my last question. You purify your samples by crystallization?
So
Or-
there's many methods to provide the molecules.
Yeah.
One of them for sure it is crystallization. For the last step, just to make sure no any kind of impurities, we
Right.
do it with column
Column.
chromatography. Yeah.
Okay. Okay. All right. I appreciate that. Thank you.
No problem. Thank you very much.
Thank you. Any other question? I think Dr. Memoli there has a question. He has been raising his hand. Okay. Well, we go with seniority too, so that's okay. Dr. Adis understands.
Thank you so much. First off, this was a great talk and very relevant. I have a couple of, actually, let's make it two questions. Number one, so as I understand, you could actually screen libraries of chemicals
Right.
because you know the targets and how they function and all that. So we have a lot of medicinal sort of plants, and this is medicinal chemistry, essentially, you're talking about. So first question, what are the global effects of these inhibitors or targets in the whole system in vivo?
Okay, so your question, what is the effect of this molecules
In vivo, in the cell?
in general in the like well we actually we did the in vivo study
Mhm.
for this for the rat who has some kind of cancer.
Yeah.
And we measure what is the effect of this compound there. But actually, as I mentioned, I don't know what is the effect, if it's going to affect on any other part of the body.
I see. We have animal models here. Dr. Foxford leads our GOAT Center.
Oh, okay.
We'll have birds. He's over there.
Oh,
And
okay.
then we have cattle, of course. But we can have other animals if needed to test some of these things in a systems level, systems physiology.
Yeah, actually, this is could be a very good start. Actually,
Yeah.
Right now, I'm working with different categories of compounds.
Yeah.
And basically, we send this to the NCI,
Mhm.
the National Cancer Institute, and they make this kind of screen for the 60 cell lines. And among them, actually, we have the breasts and leukemia very active for this compounds. It
I
will
see.
be actually a good start if we can do something like this.
Yeah.
As our next step.
This
Like
could,
we have the compounds and we need kind of that's why I didn't make more collaboration with people here, because I need to see
Yes.
someone here before I ask them. So this is a good option, but it will be my last option.
I got it. Yeah. So this could be very good NIH proposals, R03 or the other one, high risk before
That's great.
R01. And my other question is that, you know, we have a lot of these settings here where it targets another very important area in cancer, that is to prevent cancer. You see, in our country, we have all these moonshot projects and everything to cure the cancer, which is much, much more difficult, if not impossible, as compared to prevention. So we have goat products here. We have agriculture, we have gardens and all that. I think there could be, if you could think about a way to assess prevention of cancers through some of those nutritional things that will essentially make person less prone to cancer, I think that's a very fertile future
Yeah.
area.
Yeah, this would be another good subject. Actually, Dr. Faris mentioned something like this, like all the foods has some compounds, and I think the way to do it is look at what kind of the ingredients of this foods, or what is the active molecules in this
Yeah,
food.
you're right. Food is medicine, and
Yeah.
there are genetic and epigenetic aspects of cancers that, I mean, the root causes of cancers can be tied to different mechanisms. And lastly, I'd like to mention that sounds like another commonality, Dr. Ferris, is that looks like you were in the cold state of Wisconsin, was where I was.
Yeah,
And cold makes you stronger. That's what made me believe. So I am familiar with
not
Marquette
really you stay
University.
at home most of the time.
Yes, so I'm
Or-
familiar
or
with
maybe-
Marquette University, and thank you so
maybe-
much.
Thank you very much. You may stay in the lab to work, maybe. So be more
Yeah.
stronger.
Stay more indoors, right? Yeah. We had a question online about, do we provide financial support to the MS students? And I answered online that there will be, but it's on competitive basis. So we have a few opportunity to support grad students, but it will be will be competitive. So if you have strong GPA, strong support letter, and good background, we will consider supporting students for the MS degree program. We have another question here. I encourage, by the way, the online to submit additional question for those of you who are online. We have a question here from the indoors. I'm going to give the microphone here.
Thank you. Thank you for your nice presentation. I am Adise. Actually, my first question has been already taken by Dr. Mamli, but I have another second question. I have seen the IC 50 values. So to have an IC 50 values, you need to have a certain assays.
Right.
What kind of assay you used in this kind of research?
Well, again, this is a part which is people in Egypt did. I might look at the paper exactly and give you what is says, but they basically compare this one to the commercial available compounds over here. What is that say? Well, I know they did the in vitro work, but with which exactly I say, I don't know. Well, or if I look at the paper, I will know.
Okay.
Yeah.
Yeah, just for your information, in addition to what Dr. Mamli said, we have also do some, we have cancer cell lab in our center. We try to do in vitro based studies. If you have an interest to collaborate, you can reach to us.
Perfect.
Yeah.
So who I should contact if I like? Who
Well,
should-
it depends what you want to do. He's the boss there, Dr. Memoli,
Okay.
he's the boss. Another boss here, he's taking care of the International Goat Research Center.
Okay.
And we have Dr. Carson, she's active on polymers and other cancer-related work.
Perfect. Okay.
And if you'd like to about agriculture and water, you can come to me. Well, any other question for Dr. Samah?
Thank you.
Like to kind of explore mention mechanisms of making things happen. Professor, we have researchers and we have mechanisms to apply for grants, including that NIH R03 is essential for generating preliminary data. You don't need preliminary data, but you already have some. And then the other one is we have seed grants through, we are going to have seed grants through our Agricultural Resource Center here. And we especially encourage junior faculty members and researchers and postdocs to apply, to generate preliminary data, to use that to apply for grants. And of course, cross-campus collaboration is absolutely critical. So we encourage that. That generates a lot of innovative approaches. And we also have a capacity building grant program that is geared towards agricultural research. And it's the next one is coming up. So I encourage you to look into that RFA and find a potential collaborator, explore ways, visit, and then you will see the next one will just amaze you what you can achieve together.
Perfect.
Thank you.
Okay. Thank you. Thank you very much.
Okay, I have a question here from online. I don't know, question or comment, but it's a comment, it seems. This is from Dr. Rahmat Atai. He said, if you need a collaboration in foods, please contact me, contact Dr. Atai. Also, I think we have also Dr. Mora, she's here. She's also a food scientist who's also working on food shelf life extension, etc., related to food science, food. So that's another opportunity for you. Okay.
Perfect. Okay.
So as you can see, we have plenty of.
Yeah, I need to expect this actually. Like, but thank you for this opportunity, guys.
Yes.
Thank you.
So I guess, do you have any other question or comment before we conclude here? I just want to remind you, please, again, about the conference, student participation, your support and participation, the REEU, the MS degree program, and also I think we need to reach out to colleagues such as Dr. Abdul Wahid here and encourage them to present to our seminar series because, you know, it's only across the street from here, from us, at least from this building, and we don't know the type of research activities he has to do. And I reached out to him because I knew, as he mentioned in 2019, when I think you were one of the recipients for postdoc, right? And graduate students, and through the Office of Research at that time. So he was one of the 4 people that benefited of a postdoc position. So I'm glad to see that work is coming to fruit, being fruitful. I wish it was continued for another year or 2 to support your research, because postdoctoral researchers are major component of a strong research program. And we are lucky here to have several of the attendees here who are postdoctoral research fellow working on our research program. With that, I would like to mention that next week we won't be having a seminar because we have the LDE, CDE, I'm sorry, I got confused between them. So please participate there. We will have hundreds of high school students who will be competing here, part of FFA programs. So your also contribution to that program is highly appreciated. With that, thank you and have a great rest of the week, and we'll see you in a couple of weeks. Have a great rest of the day.